Scientists have demonstrated that life can persist in states once thought incompatible with survival, not by defying biology but by exploiting its most extreme limits. Recent discoveries show that certain organisms can remain viable across immense spans of time by either entering total metabolic suspension or by slowing their biological clock to a near halt. These findings redefine the boundary between life and death, suggesting that biological time is a flexible variable rather than a constant. One of the most striking examples comes from the Siberian permafrost near the Kolyma River, where researchers identified a previously unknown species of nematode, Panagrolamus kolymensis. Radiocarbon dating of surrounding plant material confirmed that the organisms had remained frozen for approximately 46,000 years, dating back to the late Pleistocene. When thawed under controlled laboratory conditions, the nematodes resumed movement and feeding, demonstrating that they have remained viable throughout their long-term freezing. The primary mechanism involved is anhydrobiosis coupled with vitrification. As environmental conditions deteriorate, the nematode gradually loses intracellular water and accumulates protective molecules such as trehalose and late embryogenic abundant proteins. These molecules act as molecular shields, replacing water to stabilize cellular membranes and proteins. This turns the internal fluid into a stable, glass-like state, preventing the formation of lethal ice crystals and preserving the cellular architecture in a state of perfect, glassified suspension. A similar but even more resilient strategy appears in the bdelloid rotifer Adenata stineri, recovered from 24,000-year-old Arctic permafrost. These microscopic multicellular animals resumed normal activity after rehydration and reproduced through parthenogenesis. Like the nematodes, they enter anhydrobiosis to survive freezing, but their survival depends heavily on an exceptional capacity for DNA repair. During dehydration and freezing, their DNA fragments into pieces that would be lethal for most organisms. Upon rehydration, specialized high-fidelity repair pathways reassemble the fragmented genome. Furthermore, through horizontal gene transfer, these organisms can incorporate genetic material from their environment, bacteria, fungi, and plants into their own genome, allowing them to patch genetic holes and acquire new resilience traits over evolutionary time. Extreme biological persistence is not limited to frozen ecosystems. In 2020, a team of scientists recovered microbial communities, including members of the Alpha Proteobacteria and Gamma Proteobacteria phyla, from ancient clay sediment buried miles beneath the seafloor in the South Pacific Gyre. These microbes were recovered from layers dated at 101.5 million years old, an environment previously reported as a dead zone due to its extreme nutrient depletion. And like the frozen organisms, these microbes were not in a state of complete dormancy or shut down. Instead, they survived through minimal basal homeostasis. By operating at the lowest known energy levels capable of sustaining life, they diverted nearly 100% of their energy toward biopolymer repair, actively fixing DNA and proteins as they decayed rather than growth or division. They were likely sustained by trace amounts of oxygen produced by radiolysis, the natural splitting of water molecules by background radiation in the sediment. When supplied with nutrients in the lab, 99.1% of these cells resume DNA synthesis, proving that life does not vanish when conditions collapse, but can retreat into states of near total suspension to wait out the ends. If you enjoyed this video, please don't forget to subscribe, like and share.